more specific to your own applications, I mean, ultimately there's going to be what's the right fit of technology for your own needs. And we have customers across all different kinds of applications and different operational needs. Some of our clients just want calibrated imagery from sensor systems, and they're going to run their own analysis in their own platform. Some of us they would like to send just RGB color imagery to us and have us run some analytics on that. We have some customers that want end-to-end full top-end service with calibrated spectral imaging systems. Then we have some clients who spent a season fumbling around with a mix of tools, realizing that spending their time clicking on images and things is not how they want to spend their time and how they shouldn't be spending their time. And they're just looking to upgrade their program and make things a lot more automated and efficient, and in a place for gathering all their data together. So part of the solution here is making sure that all those aspects of getting the right metrics, getting the flexibility of operations right, so it actually works for what your needs are. And we are really working with clients across that entire spectrum, from those that want to collect their own data and let us run analysis, to those who want full service completely and just want to look at results at the end. So let me jump into a handful of common usage examples here. One of the first ones is about looking at emergence statistics, and this is important for a range of applications. But before I dive in too much, just a quick orientation here on what you're seeing on the screen. So this is our cloud platform portal, and the view you're seeing is on the left is a satellite background image with a data layer on top of that. That's this kind of green square pattern right here. In this particular example, there are multiple data products available. There's emergence fraction, plant count, plant size, population density, which happens to be the data layer that's selected at the moment, and then weed count. In the bottom left, there's a histogram of population density for each of the individual plots within this trial. And you can see in each one of the squares within that data layer is an individual plot within the trial. And then on the right is a high resolution image of just a single plot within that trial. And there's a flag on that that's giving you the actual data on that particular plot and where it's located. So a couple of things you can kind of zoom in here. First of all, you get what's the statistical distribution of emergence across this entire trial. But the ability to take a look at an individual plot, and here what you're seeing, this happens to be corn, but you can see through a combination of the right amount of spectral resolution and spatial resolution, you can start analyzing and identifying individual leaves, and that enables us to count individual plants and size them. And what you're seeing here in the green are the detections of individual corn plants. And what we're also detecting, and maybe it's a little bit more difficult to see, but highlighted in red are weeds, basically, which are plants that are not part of the trial population. So, like I said, through the combination of the right spectral resolution and spatial resolution, we can get in and do these types of measurements. And this happens to be an example for corn, but we've done this for a number of different crop applications too, but really characterize how emergence is developing in a trial. And through a lot of ground-based validation, we've seen that these measurements are accurate to within about 2 to 3% when collected within the right window of a stage of growth. And also, there's a few data products here: emergence fraction, vegetation fraction, which get to more canopy coverage. Those are metrics that aren't really observable by humans at all. So it's an added layer of information. And one of our clients made the point recently that a single drone can replace the labor of about 80 people in the field. So collecting this information traditionally is very time consuming and laborious task, but this process automates a lot of that. So, a more in-depth look at how emergence is developing and done more efficiently than what's been done traditionally. And I'll move on to the next example here. So another common use case is in looking at stress conditions in managed stress environments. So This is also an example of a corn trial. And here, what we've introduced is time histories for a particular trial. So you're kind of looking at the same layout of information, but you'll see now next to the histogram, there are a couple of line charts. And these are individual measurements for the selected trial here, which has got a white box on it, over the course of the season. So the red curve is looking at vegetation fraction or canopy coverage over the course of the season from just after emergence all the way until the end of the season. And the other curve is basically a proxy for chlorophyll content. And what happened in this particular trial is they introduced a managed stress variable. And immediately what you see is that basically a wilting of the canopy. You see that in a decrease in vegetation fraction here. And while chlorophyll development basically leveled off for some period of time, and then after treatment recovered quite quickly. So these types of examples are, again, looking at how developmental inputs respond to managed stress conditions, and the ability to accurately measure plant development over time and make these comparisons gives you a granular new look at how plants are responding to the environment, which is valuable. So the third example here is similar to the last, where we're looking at how basically how effective are our inputs. And in this particular case, you're looking at a similar trial here, but in the bottom right is of the screen is a comparison, a zoomed in portion of the trial on a given date, and then on the right were the input prescriptions. And basically what we're looking at here is are the trials responding as expected to the developmental inputs, and looking at that relationship. So, the combination, these are just a few examples here, but they're relatively common, and they span, from breeders to crop protection and crop nutrition applications. But these are just a handful of examples about how this technology may be used.